Okay. Hi, everyone. Welcome to the RareLink. Our project is about using deep learning for detecting genetic interactions within rare disease. Here are our group members.
Yeah, I'm Shivam.
I'm Aaron.
Hi, I'm Shichen.
And David is not here right now.
Yeah. First of all, I want to do the introduction about our project. As we all know, in the current era, the existence of a rare disease still brings great suffering to patients and families. As a result, there are still many questions to be explored around the genetic architecture of a rare disease. Here I have an example. There may be about 50,000 people living with EB around the world. Furthermore, for per year per patient, the range from the 1,500 to over $40,000 is burning to the U.S. healthcare. However, the treatment is very limited. First, we have to understand that one of the main challenges in the genetic structure of a rare disease comes from the pervasive genetic interaction of phenomenon known as epistasis.
So, as highlighted in the previous slide, these rare diseases can be very complex, and genetic interactions between different genes and pathways can further complicate our understanding of these rare diseases. For example, cystic fibrosis is affected by the TGFB1 gene, and different variations in this gene can influence the severity of this specific disease. Additionally, retinitis pigmentosa is another recessive disease in which mutations in two different genes together cause that disease, and sickle cell disease can be affected by different levels of hemoglobin, a biomarker that can be affected by a variety of genes, including BCL11A and HBS1L. And again, the levels of hemoglobin can determine the severity of the specific sickling phenotype. So, all in all, different dominance relationships between these genes can make diagnosing autosomal recessive diseases very difficult.
Yeah, so we created RareLink, which is a deep learning method to detect genetic interactions to try and get a handle on this problem of epistasis and genetic interactions. So we've all heard about things like AlphaFold that have come out. Basically, a lot of these large language models are they're being used for DNA sequences and protein sequences, and they've been very helpful within the field of computational biology. So these methods are actually really useful for detecting genetic interactions, and this is how we leveraged it. So first, we're only looking at two variants, two mutations, and trying to understand the interaction between both of these. And what we can do is we can take a sequence of DNA without any mutations. Then we can make the first mutation, pass that DNA in through our machine learning algorithm. This is a GPN MSA, which is a DNA large language model. And then we can not only look at the activation of the neurons within this large language model to see potentials for interactions. We can actually take one of the intermediate embedding layers, and pass it through this thing called a sparse autoencoder, which allows for interpretable features. And then we end up making some sort of epistasis score using a transform on the features from the sparse autoencoder. So the sparse autoencoder is really useful because it actually takes all these complicated neurons that are firing within the large language model and making it very simple of just a few things are firing. And these few features that are firing actually correspond to um, actual biological elements within the sequence. So we'll pass in the sequence with the first mutation, pass in the sequence with the second mutation, and then we'll pass in the sequence with both of the mutations together. And what we can do is we can look at how these interpretable features change between just the first, just the second, and both of them together. And we're saying that there is actually some epistasis if there are features that were turned on for the first variant that actually turn off when you introduce the second variant, or vice versa. And that shows that you can't actually predict the phenotype of both of the mutations together just by taking the phenotype of mutation 1 and mutation 2. And this GPN MSA large language model is trained on ClinVar, which is a large database of variants that impact disease, including rare disease, as well as OMIM. And we included Evo as another example. Evo is just a different kind of large language model. We think it'll be more powerful than GPN MSA, so we plan to expand onto that in the future. Um, so we wanted to test whether RareLink really works or if it's useful and what the general framework would be like. So we ended up using this gene LMNA, and we were using RareLink to predict the loss of function genetic interaction between two disease implicated variants in LMNA. So this is exactly what we were talking about before. We have variant 1 and variant 2. And we get some sort of functional score, which is just the net amount of feature activation from our sparse autoencoder. And we see that variant 1 reaches about 57, variant 2 alone reaches about maybe 76. But then you put both the variants together and you find that the variant pair isn't actually some sum of the individual features from variant 1 and 2. We find that the variant pair is actually less than variant 2, variant two's feature score. And the idea here is that you don't have as many features turning on as if you just had variant 2 alone. So the presence of variant 1 is impeding whatever function variant 2 has on the actual DNA's pathogenicity. So what we did here is we decided to say that variant 1 is actually making variant 2's function kind of neutralized. So it's kind of a loss of function. So we're taking this final value from the variant pair, and then we do a simple transform to get it ranged between -10 to 10, which will give us some notion of a strength of epistasis. And we're saying that if that epistatic score is much less than 0, we're saying loss of function. If epistasis score is about 0, that means there's no interaction. And if it's much greater than 0, we're saying it's a gain of function interaction. Now, we still need to look at many, many more genes to see, first of all, is this the right methodology for actually taking the sparse autoencoder features and turning that into an epistasis score? And we need to do some sort of clinical testing or some sort of laboratory testing to see, okay, are the variants, when we have two variants that have some epistasis score that's very high or very low, does that actually correspond to anything biologically? But this is our first kind of proof of concept.
Okay. Thanks for the Shivan's explanations. Our RareLink also have a web application to help clinicians. We have three sections. The first section is a clinician can pick one gene from a 10 specific gene sections. The second section is the clinicians can choose one or two variants. As a result, it will generate one disease report to help clinicians to make more better clinical decisions.
So for future directions, we'd like to focus on scalability. So the ability to use our dataset for all localized rare disease variant pairs in the human genome to really flesh out the range of variants that are accessible on our RareLink platform. We also want to focus on biological interpretability. So using these sparse autoencoders that Shivam described in the earlier slides to detect biologically relevant features of DNA sequences. And the question we're getting at here is, can we detect new disease mechanisms and potential therapeutic targets using these sparse autoencoders? We'd also like to refine our metrics and see how valid our epistasis metrics are, if the interactions that we're detecting are real, and if we're able to compare these metrics to data from CRISPR screens and other in vivo methods to get as accurate a picture as possible. And finally, we'd like to flesh out a user-friendly platform to see how clinicians will use RareLink to get parents a better diagnosis in real life, and if we can improve the workflow in rare disease care. we aim to do user interviews across different providers and researchers to achieve this goal. Finally, this is our team. We really enjoyed the hackathon, and thank you for sponsoring and hosting.
Yeah, thank you so much.
Mhm.
Happy for this opportunity.
Yeah, thank you so much.
All right. Okay.
Sweet. Okay.
Yeah.